Okay, so first of all, I'd like to thank the organizers for putting together this nice meeting and giving me a chance to present some of the work that we've been doing. So I'm Mark Becker. I work in Clare Boiler Club in Manchester, and I'm going to show you how we incorporated AlphaFold into designing a new model system to study one of the proteins that we're interested in. But before I jump to that, I'll just give you a quick overview of what we do. So in our lab, we're mostly interested in extracellular matrix proteins. And for those of you who didn't have much exposure to the extracellular matrix, is this complex three-dimensional network that is formed in between cells. You can see it quite nicely in these SEM images collected on fibroblast cell culture. And it's essentially a very complex three-dimensional network of large macromolecules that are important for the structural integrity of tissues, but also for regulation of cell signaling and mechanosensing. And as you can imagine, between different tissues, the composition varies quite a lot. But generally speaking, you have proteins like elastins, fibrillar proteins, and one protein that I'm going to focus on, which is collagen. And just maybe a little pub quiz fact to keep from this presentation: collagen actually accounts for about 30% of all the protein in your body. So that just goes to show what an important role it plays. So we are particularly interested in collagen 6. Collagen 6 is found in most tissues throughout the body. It's a so-called microfibrillar collagen. So unlike collagen 1 or 3, it doesn't form these massive fibrillar structures that some of you might have seen before, but it forms smaller so-called microfibrils. And it consists of a series of van Brabant factor A domains, which are small globular domains, and they're connected through Gly-XY repeats, which in the final assembly form a triple helix. And this assembly mechanism is actually quite a complex one. You have 3 different chains coming together to form what's then called the monomer, which then dimerizes, tetramerizes, gets secreted out of the cell where it forms these microfibrils. And just another thing to highlight how complex this system is. These are all of the chains that exist. Humans don't have the alpha-4, but essentially all of the microfibrils will consist of any combination of these chains. So it's quite challenging to work with. And the traditional way to get hold of collagen 6 for really any types of studies is to isolate it from tissue. And usually that's done by extracting it from bovine cornea. And you can do that reasonably well. This is work that one of the postdocs in our lab has done. And you can see this negative stain image. You can see this microfibril with a very typical double beaded region, which is formed by the van Brabant factor domains. And he was actually able to prepare cryogrids with this and send it for a cryos data collection, where he got just under 30,000 images, I think. And he was able to get these really nice 2D classification of the double beaded region and reconstruct it to what is about 6 to 7 angstrom map. And you can quite clearly see these globular regions. These are the individual van Brabant factor domains. And you can even see the collagenous region running down through the center. And you can even start to dock domains into this density, which is what he's done here. However, the problem is this prep will be a heterogeneous mix of all the different chains because they are expressed in all the tissues, so we can't control for that. Also, the domains are quite flexible relative to one another. So you can, if you do a flexibility analysis, you can actually see them rotating around the core. So all of these are things that we can't really account for with tissue-derived collagen, but that hold us back from getting higher resolution information on this. And so we asked ourselves, can we make a model system where we can control the composition, where we can just make it in the lab, and we can hopefully account for flexibility by just having something that's smaller, so you have less moving parts. And our inspiration from this actually came from another model system called CP3, which has been used for about 20 years now to study collagen 3 assembly. So collagen 3 is one of these fibrillar collagens. It is expressed with prodomains, which are proteolytically removed. And people have made various constructs to study these processes, and they have found that they are quite well behaved. So this is a crystal structure that was made on a shorter version, and other groups have shown that you can use these constructs for cleavage assays and other in vitro studies. So it's quite a nice model system, and we thought, can we recreate this for collagen 6? Now, obviously, the first problem is you've got 3 different chains just to start with, and then so we decided just to replicate the collagen 3, we take the C-terminal domains of the alpha 1 and 2, include some of the GlyX-Y region to hopefully get a hint of this triple helix. And then we moved on to alpha 3, and the first problem was alpha 3 has these extra domains. So we were like, do we keep them? Do we remove them? Keeping them gives us more information about the system, but it might also make it more difficult to study for cryo-EM. So we settled to make 2 constructs, one shorter one that just resembles the alpha 1 and 2, and a longer one with the extra domains. And as we were going through this process of designing it, we thought, let's just see what AlphaFold thinks this is going to make if we just give it the sequence for the constructs that we're planning to make. And this is what it came up with. It's quite a nice prediction. You can quite clearly see the individual domains in the triple helix, and it scores quite well in terms of the PTM and IPTM, which granted isn't too surprising because there are crystal structures for the individual domains, but it's still good to see that AlphaFold can make a trimer out of it. And then we started looking at it and saw some interesting features. So you can see this triple helix here, which is quite characteristic for collagens, and you can see it's quite rich in prolines as well. And as we were going through the structure, we found this nice network of disulfides that's stabilizing loops and helices, both within the individual chains, but also across the chains. And looking at the center of the protein, so that will be down here, we found this 3 helix bundle where all 3 chains come into contact, and they're actually also disulfide linked. And so we thought maybe this is the recognition motive that ensures that in nature you always end up with a 1, 2, 3 assembly, or 1, 2, 5, 1, 2, 6, but never a homotrimer. So encouraged by these results and seeing what we might be able to achieve, we then went ahead and made these constructs. So I focused on this shortest C1, C2 fragment for the alpha 3, where we instantly ran into another complication in that there is a furan site in front of the C2 domain, and it seems that in our expression system, this furan site is indeed cleaved in some cases. Nonetheless, we were able to purify the complex, and we put it through 3 different affinity purifications, making us reasonably confident that we have indeed a heterotrimer. So then to be sure of that, we did SAC-mols analysis, and this gave us 2 peaks, which near perfectly correlate to what you would expect to a heterotrimer with or without this C2 domain. So we see that we have both species in there, and we can't really separate them, but we are reasonably confident based on the mass that it is a 1, 2, 3 trimer. And just to check this, to get like more of a sanity check for this, we then decided to do mass photometry. So given that it's a relatively new technique, just briefly the way it works, you essentially have a glass slide, you have light shining on it that gets reflected, and your sample goes on top. And the idea is whenever a molecule in your sample touches down on the glass, it will scatter the light, and the scattering of this light correlates to the size of your particle, and the size of your particle correlates to its molecular weight. And then you can use a weight standard to convert this light scattering back into mass. And so we've done this for our sample, and this is the sample before it went down a SEC column. And what you can see is the majority in the sample correlates to this heterotrimer, although interestingly, we don't see a difference between it having the extra C domain or not. Although if you look at the AlphaFold models, you can see that the shape of the protein actually doesn't change that much if you remove the domain. So maybe that's also one of the limitations that you see with the mass photometry here. But you also see that we have the free C2 domain floating around in the sample, which luckily we were able to remove doing size exclusion. And then lastly, we collected small angle X-ray scattering data on this. And what's quite nice when you use SAX is you can essentially take any PDB file and simulate SAX data for it. And then you can compare it to your experimental data, which is what I've done here. So the red one is the AlphaFold model. And it gives you a chi-square. This one's 2.6, which for fitting PDB files to SAX data is actually quite a good score. So that tells us that not only we have a trimer consisting of all three individual chains, but it actually looks like the AlphaFold model. So we know it looks like we want it to look, and we're not just purifying something completely artificial. So based on all of that, we then decided to start going into EM. And first of all, as like another quality control, something that we do as a standard usually in our lab is we did negative stain, and you can see these very distinct particles. If you look over this image and just over the grid in general, you can see that it's quite a homogeneous mix. So that's always good to start off with something relatively homogeneous before going into cryo-EM. And we actually did a reconstruction of it. I mean, it's a negative stain, it's like 20 angstrom. But what's quite interesting about this one, if you compare it to the AlphaFold model and simulate a 20 angstrom map around it, you can see that these two maps are really similar, and you can see quite easily how the AlphaFold model would fit into this density. So again, it shows that the AlphaFold actually did quite a good job at predicting what the system would look like. So then we went ahead and did a screening session on our glaciers and collected a few hundred images. And we got these really nice 2D classes out of it where you can see the individual domains. And then what was quite interesting, as I was playing around with the AlphaFold model, I realized that if you generate 2D classes from that model, they are extremely similar. And I promise these are not handpicked. I just took like 9 classes from each and put them next to each other, and they are very similar. So again, you just see that AlphaFold really did a good job at predicting how our system would behave before we even started doing anything in the lab with it. Now, as I was working on the C1, C2 trimer construct, our postdoc was working on the longer alpha-3 construct, where we had actually knocked out the furan site to account for getting a mixture. And that's reflected in the mass spectrometry data. So you basically mainly get this species here. There is this larger molecular weight species there, which we don't know if it's the trimers starting to dimerize or if it's just aggregate that's in the prep. But we were able to remove that with size exclusion and get a quite clean prep. And so we decided to send that sample for data collection at EBIC. And this is just some of the parameters we used. So we're lucky enough to collect on the Cryos 3 with the Falcon 4i and the energy filter. And over about 24-hour session, we ended up with like 11,500 images, which I think just shows how fast data acquisition has come, especially with the new sensors. So we were quite excited by this and instantly started diving into the data processing, but very quickly realized that as nice as it is to have this clean sample and as nice as it is to have almost 12,000 images, we just weren't able to get decent 2D classes. And I'm sparing you the 3D reconstruction. It doesn't look great, but as you can probably guess based on the classes. And we realized this is basically because this larger construct still has too much intrinsic flexibility. In fact, there is some literature indicating that this C3 domain is disordered. If you make an AlphaFold model, it can't really make any sense of that region. And so it's despite all of the promising data at the beginning, this just shows that this system is still limited. So we're now planning to send the shorter construct. We're working on one where we have knocked out the furin site in that as well, just to get a really clean sample. And we're preparing to send this for a cryos data collection, which, based on the classes I showed you before, we're quite confident that this should go to quite high resolution, which hopefully will then help us to answer some of the questions around how collagen 6 assembles in the matrix. So just as a quick summary / outlook takeaway message. So we used AlphaFold in this to design constructs before then making them recombinantly, and it was a really helpful tool for us to understand how to set cutoffs and things like this. When we expressed them, simple co-expression in the same cell culture, they did instantly form heterotrimers. And analyzing the AlphaFold model, we do see some very interesting features that might be relevant for our biological question. But nonetheless, we've seen that the larger constructs, there's still too much intrinsic flexibility. Collagen 6 also has various post-translational modifications, and they are still holding us back from getting the high-resolution data we want. So we're working on modifications for these constructs to make that easier. And I guess the take-home message from this talk that I wanted to show you is just AlphaFold can be really powerful tool if you know how to use it, but it can't account for all other challenges that I guess all of you in cryoEM are well familiar with in terms of flexibility, because AlphaFold can't really predict these sort of things. So with that, I just like to thank the people who helped me with this. Of course, my supervisor, Claire, our postdoc, Al, who's worked on this collagen project for far longer before I got involved with it, and also especially our technician, Rana, who's been really, really helpful with all of the lab-based work. And then the facilities, the EM core facility and the biomolecular analysis facility in Manchester, and at Diamond, the B21 beamline, where we collected the SACS data, and the EBIC as well. And thank you for your attention.